the cytoskeleton, and that is basically we have three types of cytoskeleton. We have the actin, we have the intermediate, and it is intermediate because in width and diameter between the two other filaments. And then lastly, the biggest one we have is the microtubule, or the MT. So basically, what is cytoskeleton? It provides the shape and movement to our organelles, and it is a dynamic protein, so it's a dynamic filament, meaning it's always moving, or ability to move, add and subtract, basically. So it's a dynamic filament that provides shape and movement, and we have three types: actin, intermediate, and then microtubule. So what do these ones do just overall? So actin is for the shape of the cell, and a good example of that we will learn about is the lamellipodium, and how it is able to, it's like a worm, and how this worm is able to move in this direction. Then we will talk about microtubule, and that is how organelles move or position themselves. That is the microtubule is how organelles move and position themselves. And then we have intermediate filament, which is for the structural component, everything in the middle, and we find that usually in the nucleus. So that is structural, nucleus, and found in the skin keratin. So after these three types, let's just talk about the overall role of the cytoskeleton. So role of cytoskeleton, and this is just a big list because you can imagine they can do so much. So what is the example of role? So we already talked about shape, the deformation or the contraction of a cell, it moving, motility, and cell division, and we have the microtubules that pull the centrosomes or centrioles, and then we have signal transfer, the cilia, pinocytosis, chromosome segregation, and then the cell envelope template. So that is just overall what is cytoskeleton, what are the three types of cytoskeleton, what they do, and what is the overall role of the cytoskeleton. So now let's go one by one into each one. So here, as you can see, just a quick example, we have the microtubules that are in green, and then we have the red actin, which is surrounding all of the cell basically, and that gives it the structure. Once again, intermediate filaments, they're intermediate in width, so they're 10 in between the actin, which is the smallest and most numerous, 7, and microtubules, which you find in the mitotic spindle, and as well as in cilia. So intermediate, once again, you find it in skin. That is how the skin cells are connected to one another using these thick bundles of intermediate filaments that are formed all the way around. And how does a intermediate filament form? It is basically a alpha monomer, and those two alpha monomers can become coiled dimer, and then you can have a tetramer, which is staggered, and we call that the rope-like, and it gives it the cytoplasmic rebar. So it's basically gives the cytoplasm its very strong, tough structure to hold the organelles. So let's go start with the first one we will do is the actin. and we learned that actin was for cell shape and movement. And they're around 7 nanometers wide. And we find it in the example would be the lamellipodium. Okay, so they provide shape, and basically they are also polar, how they form. And we have two that are polar. So microtubules are also polar. And intermediate is the only one that is nonpolar. So they are polar, meaning you have two ends. So if you have a positive end and a negative end, so you can have actin is ATP bound. So, and then on the other side you have ADP. So the side that you have ATP is more stable, where growth is happening. Basically, it's growing on this side. And on the other side, it is unstable, or it is losing its components. Whereas microtubules are GTP bound. So actin A, ATP, microtubules, GTP. So how does actin form? Actin is a two-stranded repeated bundle. And it is polar due to the ATP and ADP, like we said. And this is the myosin tract, the sliding filament mechanism. That is what's used in muscle contraction. So in muscle contraction, actin is the track for myosin, with the sliding filament mechanism. So this is what we call the muscle of the cell, or the cell muscle, or the contractile unit. So actin is the muscle of the cell, or the contractile unit. And that's the way actin works, like I showed above. We call that treadmilling. And so how treadmilling works is that once again you have two ends. You have the positive end and the negative. On the negative side you have the unstable ADP, which is losing, and on the positive end you have the ATP, which is growing. And that is how the muscle is able to, or the myosin is able to move along. It keeps moving as this treadmilling of actin. So if you have myosin, it would basically keep moving here. This is my stick figure. So he keeps moving along because the length keeps getting increased. So once again, your myosin fibers are your thick fibers in the muscle. And I will make another video for muscle contraction. So myosin walks on actin, which is thin filaments. Like we said, 10 nanometers wide. And the way more specific example is your pseudopod formation, or how lamellipodia move. So how that works is similar fashion, is that you have this lamellipodia, basically worm-like structure, and he wants to move that way. So what starts happening is that you have positive actin on one side that is polymerizing, and it attaches to a newer structure or grows in this direction, whereas on the other side, your actin myosin slide and they contract. So you have, so you have attachment on this side, whereas you have contraction with this actin and myosin. And so in that way, after contraction and attachment, this lamellipodia is able to move along. So all of this is due to the actin polymerization. So that is everything we talked about of adding with on the ATP end. Okay, so next we will talk about the intermediate. After actin, which is the smallest in width but the most numerous or the longest length, we have intermediate, which is middle in width and gives it that cytoplasmic rebar or the structural shear, we call that. So the most important is that it is intermediate in width and provides the structural shear. Also, we'll call it the cytoplasmic rebar. So it is basically found in the skin keratin. In the skin, it gives it the strength or the glue, as well as connects the skin cells. We also find this in the in hair, feathers, claws. So you have two categories of intermediate filament. You have cytoskeleton, or sorry, cytoplasmic, and nuclear. So in cytoplasmic, you have the two types you can find in the keratin type, as well as the neurofilaments. Whereas nuclear, you have the nuclear lamins. So what is the purpose of intermediate filament? It is to provide us with this rebar or this strength, structural strength. Due to the structure of the intermediate, which is a coiled alpha helix. So it is a tetromere of an alpha coil, basically. Unlike actin, which was a two-strand repeat, but was also polar, whereas intermediate is the non-polar. Whereas actin and microtubule both are polar. So how does this form? You have the alpha helical or alpha helix, which gets coiled. So you have a coiled alpha helix. Then you get the staggered of the two alpha helixes, or coils, staggered of two coil, and that gives us our cytoplasmic rebar, or the shear resistance. So basically it is a coil, two coils that make up the coil. Sorry, this is horrible, but you get the point. It's nonpolar, and you can determine specificity by looking at the domain end, or the end domain. So you have a varied end domain, and that tells you about the intermediate filament. Once again, it gives us the cell shear resistance. We call that the cytoplasmic rebar, or the toughness. We find in things like our hair, feathers, claws. So you have the keratin type, you have the neurofilament type in cytoplasmic, and then the only one is in nuclear is the nuclear lamin. If we go back to some images and look at this, so we have here we have our outside, which is the actin, which is the actin that gives it the structure. You have the microtubules in the middle, basically, that stretch from that gamma tubulin. And here we have the three. We have actin, which is the smallest one, so it's 7 nanometers. Intermediate is intermediate in its width. Microtubule is the largest one in width. Actin gives it the cell shape and movement. Intermediate provides the strength, and microtubule positions the organelle. These are the examples how lamellipodia contract with that ATP polymerization, where ATP is added on one side but subtracted shrinks on the other, whereas microtubules, mitotic spindle, grow and end on the same grow and shrink on the same end. Here we have intermediate filaments that connect the cells. And here we have first how an actin forms, or intermediate filament forms. So it also is alpha helix, like the actin in monomer. You get the coiled of 2, and then you get the staggered tetramer of 2 of those coils, and it is nonpolar. Once again, you have 2 types of intermediate filaments. You have the cytoplasmic and nuclear. In cytoplasmic, we find the keratin. We find in the epithelial, some in connective tissue muscles, and then in nerves we have the neurofilaments. The nuclear lamin is the only one found in the nuclear. And going back to actin, you have the minus end, which is ADP bound, and you have the positive end, which is ATP bound. And that is how actin treadmilling happens, is that you have growth on the positive end and you have subtraction or shrunk on the minus end, and that is shown here. So we have actin treadmilling, whereas we have dynamic stability, which is for microtubules. So actin's treadmill, whereas microtubule has a dynamic instability. And all that means is that in actin you have the growth that happens, the actin is getting added only to the plus, to the ATP bound end, whereas on the minus end you are losing the actin monomer, and that is ADP bound. So actin, ATP, A's, the treadmill, whereas microtubule is GTP, and they have dynamic instability, meaning it grows and shrinks on the same end. So you have GTP bound on the plus end, and you have growth, whereas you are GDP bound at the same plus end, and you are shrinking. So that is dynamic instability, and once again, how lamellipodium are able to move the actin cortex is by you have actin polymerization at the plus end, whereas you have myosin sliding filament that happens on the minus end, and that contracts. Lastly, we have to talk about the biggest one, which is microtubule. And microtubules help the organelles move, as well as in mitosis. So they're essential. Their most important job is for mitosis segregation. They are 25 nanometers, so they're the biggest ones. They're also polar, but they are GTP bound. And so let's talk about the structure of microtubule. It is an alpha and a beta tubulin. Unlike actin and intermediate, they were both alpha. So these alpha and beta tubulin, you can have 13-strand alpha beta dimer, and that forms your microtubule rope, basically. So once again, it is polar with the GTP side, which is stable, just like ATP and actin, and GDP, which is unstable. But it happens on the same end. So if it is GDP bound, it is unstable and will shrink. GTP, it will grow, but at the same end, unlike actin, which happens on the opposite ends. So the use of microtubule is for, once again, mitotic spindle, vesicle tract, and movement with cilia and flagella, usually found in the basal bodies. Next, we will talk about how in mitosis this happens. So the microtubule assembly happens at the centriole, and from the centrosome you have a gamma tubulin from which the microtubule is able to grow in the center, and microtubule grows in all directions. So once again, we talk about lastly dynamic instability of microtubules. And what that means is that if it is, let me draw a better picture. So if it is GTP bound, it will grow. And if it is GDP bound, it will shrink. But this happens at the same end, even though it is a polar. It is a polar cytoskeleton. So lastly, we just have to talk about the drugs that can inhibit microtubules or break microtubules, and these are important in cancer because you can inhibit mitosis. So you have two, this one and vinblastine or vincristine. They both prevent polymerization, so you do not have the growth, whereas Taxol is able to stabilize and prevent shrinkage. So Taxol prevents shrinkage, whereas the colchine and the vinblastine or vincristine prevent polymerization, so you do not have mitoses. Another side topic is that you have a microtubule or tubulin homologue that you find in bacteria. So you have a bacteria example of a microtubule type structure. It's FTSZ, and we are able to see through a microscope how it helps bacteria in cell division by separating two bacterial cells. So it separates two bacterial cells, and that is your FTSZ, just like a microtubule. Lastly, the most important concept is that the energy transduction for microtubules, basically. So this is a fact to remember. Energy transduction is from GTP hydrolyses, from a kinesin powered motor or kinesin motor, and not from the microtubule. So the energy transduction is from a kinesin motor, and that is both in mitoses and moving of the cilia and other uses. So in mitosis only you have both use of dynamic instability and treadmilling, but the energy is always from the kinesin motor. So that is everything for cytoskeleton. So we have cytoskeleton, which helps us in shape and movement. We have three types: actin, intermediate, and microtubule. We have actin, ATP, which helps lamellipodia move. Intermediate is intermediate in width and provides us the structure in nucleus and skin keratin, whereas microtubule helps us in segregation in mitoses as well as organelle movement. And these are the role of cytoskeleton. If we put all of them together, what they're able to do: actin, once again, is the polar, whereas intermediate is nonpolar, and microtubule is polar. Actin is ATP bound and is able to grow with treadmilling, whereas microtubule is able to grow with that dynamic instability with the help of GTP. Once again, this is the muscle contraction. Actin is a track for myosin, how myosin is able to use the sliding filament mechanism, so it's able to move at the ATP end. Whereas in intermediate filament, this is mostly for structural rebar or structural strength to our keratin, neurofilaments, and nuclear lamin. And then finally, the biggest one is your alpha-beta tubulin, which is microtubule. It is polar, but GTP and GDP, they help in growth and shrinkage at the same end. And then finally, we have two types of drugs that are either able to prevent polymerization, addition, or prevent shrinkage, subtraction. And then we have a homolog in bacteria, which also helps bacteria cells divide.